This is the second part of the first lecture, and we're going to talk about studying the brain. So, overview just quickly before we jump into it. This section of the lecture, we're briefly going to talk about the brain itself, and for some of you, it is not going to contain enough detail. And if you would like more information about the brain in general or parts of it, you're welcome to contact me and get more information, or you're welcome to look into it further yourselves. For others of you, this is going to seem completely overwhelming, and that is okay. Please don't stress overly much, as most of the semester we will not really deal with the nitty-gritty of brain regions, but rather just higher level functions and that they're correlated with certain brain areas. Not so much that you need to know the intricacies of, for example, the thalamus has 36 nuclei or something like this. That being said, it is important to at least know the general layout of the brain because it is important to know when talking about any type of cognitive science. All right, so the human brain. Here is a picture of what the brain looks like. Please note that this little blue sheath covering it is probably a product of how they've cured this brain. This brain is prepared for people to touch at an exhibit, and so it has actually been cured. But this is a live human, well, not alive, a human brain that is no longer living. Now, the brain itself contains approximately 86 billion neurons and between 86 and 860 billion glial cells. Now, please be aware that in your textbook it is said that there is a trillion neurons. This is incorrect. As of most recent evidence, an Argentinian scientist centrifuged a brain and used the mass of nuclei in it to establish how many neurons were actually in the brain. And commonly it was actually said to be about 100 million, so my guess is the book has typos in this regard, and that has been now down estimated to 86 billion, and that is our current best estimate. Now, neurons are the cells that actually fire or have electrochemical activity and are largely responsible for all of our thinking and feeling and sensation. Glial cells are all around all of the various neurons, and they perform many functions, some of which we are still discovering today. But some of the primary ones are glial cells encourage the correct direction for neurons to grow. For example, they are what's responsible for establishing your neurons being able to connect from your brain to your fingertips. Further, they form specialized sheets, which we will talk about in a little while, on the long portions of your neurons that allow them to conduct signals at faster rates. They're also responsible for holding the various neurons together in your brain in correct orientations, along with certain cleaning and maintenance functions, and we are likely to continue to discover more as time goes on. The brain itself weighs approximately 1.5 kilograms, or between 3 and 4 pounds, and it is the source of all behavior, memory, and sensation. Now, just a quick note here, you may or may not be someone who is subscribes to some form of creationism, and that is totally fine. That is your business. At the end of the day in this lecture, we are going to assume that the brain is the source of these three things, and there is no additional material or special sauce, or even soul for that matter, that contributes to the behavior of a human being. Ultimately, that is speculation. It cannot be proved one way or another, but for the most part, most cognitive science moves forward on the assumption that the brain is the driver of all behavior. Now, can you disprove that there is some immaterial part that drives all of it? No, not really. But please be aware of the homuncular fallacy. There cannot be a little you sitting inside your head that drives everything else, because that in turn would need another smaller you to sit inside the head of to drive its circuits, which would need a smaller you to drive its circuits, and so on ad infinitum. And you could only do this so far because eventually you would get down to atoms, and individual atoms ultimately cannot be divided unless you have particle accelerators and so on. I don't mean to get into a debate about theology here. It's just wanted to make the note that regardless of your own cherished beliefs, which are totally acceptable on either side of the spectrum, it is important to note that most of what we will talk about will be under the assumption, at least, that the brain is the final arbiter of all of our behavior, memory, and sensation. The largest divisions of the brain fall under three categories: the hindbrain, the midbrain, and the forebrain. So the hindbrain sits right above the spinal column and is largely involved in all primary life functions, such as breathing, heart rate, or blood pressure, and it ultimately regulates all of these processes. It is also something that is common across almost all animals that exist today. I'm not 100% sure about the avians, as they have unique brains, but all reptiles and mammals have some portion of their hindbrain. Another portion of the hindbrain is known as the cerebellum, which we will see as we go down through pictures. It is interesting to note that the cerebellum actually contains the majority of your neurons, something to the tune of 60 to 65 billion of them, which is pretty unbelievable because it is smaller than the rest of your brain, which contains less neurons than itself. But it is a very unique structure that has been associated with all types of different behavior. Originally, it was thought to just manage certain time-based behaviors, but it has been shown to be incredibly active in not only balance, but integrating various sensory information and ensuring that our thoughts flow smoothly. The midbrain is a relatively small area, and it sits sort of between the hindbrain and the forebrain. We won't talk about it much beyond that it is involved in attention and certain reward circuits pass through it, and it does direct certain types of specialized motion like that of the eyes. Finally, we're going to talk about the forebrain for pretty much the rest of this lecture, and with the exception of the cerebellum, it is going to be most of what we talk about, and most of the phenomena that we discuss will have some root in this forebrain. The cortex. This is probably the most obvious feature of a brain, and when all of us think about brains, we tend to think about that squiggly surface of it, and we are actually looking at the cortex when that's what we're looking at. Now, the cortex itself is actually a sheet of cells, and it ranges in thickness from 2 to 6 millimeters. That is probably less than 1/8 of an inch up to 1/4 of an inch approximately. And if we were to flatten it out, it takes up approximately 2 square feet, which would be the same area as a circle with a diameter of 19 inches, or a pizza, which I think is a fun analogy. The book says that's an extra large pizza, but my guess is that depends on where you get your pizza. Now, it is a fairly large flat surface, and it is stuffed into our relatively small heads in comparison to that surface. And there's a lot of other stuff in your head too, and this is why it has this folded appearance. And just an extra part to know and add to your knowledge of random trivia facts is that the degree of wrinkles on the surface of the brain is actually a better indicator of intelligence across the various mammalian species than the actual size of a brain. This is why, for example, a rat is considered to be smarter than a manatee, even though a manatee's brain is far larger than a rat's. A rat's brain has far more wrinkles than a manatee's brain, which is actually almost smooth, which in some degrees makes sense for manatees because they pretty much eat and go up and down in the ocean. And unfortunately, this is probably why they get hit by so many boats. Well, not entirely. That's somewhat victim blaming. They also get hit by boats because human beings don't care about most of these creatures and run huge boats without paying attention to where those poor sea cows are. Regardless, that's my biases coming in. One last piece, these wrinkles of the brain are called convolutions. Now there are some important convolutions, and the three are a longitudinal fissure, a central fissure, and a lateral fissure. You will oftentimes see these referred to as sulcuses or sulci, where one could say something like the central sulcus. Now, we'll just, I don't think I can turn on a little pointer in this yet, but let's see. The longitudinal fissure, if you look at the topmost picture that's presented in color, is running down from front to back. Now, the pink region is towards the front of the brain and the blue region is towards the back of the brain. And this divide across the middle is what is referred to as the longitudinal fissure. It divides the brain into two halves, and each half from front to back is called a hemisphere. So you have one right hemisphere and one left hemisphere. The central fissure can be seen dividing the pink from the green region, or in the second picture is outlined in red. This is often considered the mark that divides the frontal from the parietal lobes, which I will show you another picture of in just a minute. Now, please note that they're not actually hard divisions in the brain. Rather, we just use these as a way to make it more convenient to talk about locations and functions in the brain. Finally, the third important convolution to know is the lateral fissure, and it runs diagonally upward and backwards along the sides of each hemisphere. So you could kind of imagine this from, let's see, your cheekbone running over your ear upwards. And it divides the frontal from the temporal lobes. Moving on, just so I can show you what the lobes look like. Here's a picture from your textbook that divides the four different lobes. You see the frontal lobe listed in blue, the parietal lobe in red, the occipital lobe in yellow, and the temporal lobe in green, along with the fissures as well. Underneath the lobes, you can see part of the cerebellum peeking out in the back. Now, the frontal lobe, or just briefly about them, the frontal lobe is largely responsible for what we talk about when we say executive processing. This is whenever we're talking about how humans are greater than most other creatures, we're talking about a lot of the functions that we get from this region. Now, just saying executive function is not enough. With most things, you will see that it is far more complicated. It is also largely involved or called the association cortex, and this just means that it is the part of our brain that allows us to tie very disparate pieces of information together. Another thing that is considered to be processed here are social norms, which are relatively abstract. The parietal lobe is oftentimes associated with spatial reasoning. It is what allows us to keep track of things in three-dimensional space, and it is considered to be one of the hotspots of consciousness, actually, that you can actually have a perfectly conscious person without most of their frontal lobe. There are arguments and debates on either side, though. The temporal lobe is oftentimes related to language processing and audition. Here is where most of what comes in through your ears is processed, and oftentimes semantic labels are considered to be processed here as well. The occipital lobe is in the backmost part of your brain, and this is largely responsible for processing visual input. Now, please note that that is a very loose guideline of what the lobes do, and it is generally referring to just the cortex of the brain, while we will now talk about subcortical regions that are technically part of these different areas, but that they have their own unique functions as well. Getting into subcortical regions. So these are the regions of the forebrain that are underneath or wrapped in the cortex itself. And these can be seen with these various little colored images with a transparent full brain all the way around it. Now, occasionally people will talk about something called the mammalian brain, and oftentimes they're referring to the limbic system. And they talk about this because the limbic system and other subcortical regions are largely involved in all of the creation of our emotional sensations and emotional processing. This is called the mammalian brain because these structures are largely conserved across most of the mammalian orders. That ultimately leads one to the conclusion that no matter what you would like to believe one way or another, the chances are that most of the mammals at least experience similar emotionality to us. They might not have the cognitive mechanisms to be able to process those feelings, but chances are for them, fear and anger and joy are very similar at the end of the day to the sensations that we experience when we are having those emotions. The first we'll talk about is the thalamus. This is seen in purple on this diagram here, and the thalamus is largely considered to be a sensory relay station. What this means is that information coming up through your sensory organs, your eyes, your ears, your skin, is largely flowing up your spinal column. Well, not for your ears and your eyes, but it flows through this subcortical region and from here up and out into the rest of the cortex. Now, The exception to this is olfaction, which actually flows through something called the olfactory bulb, which is a different subcortical region. Now, it is important to note that this is not its only function. The thalamus is very widely connected to the entirety pretty much of the cortex, and it is involved in all manner of things. It actually has multiple sets of nuclei, which are small clusters of specific types of neurons, and some of them we still don't even know what they do. Beneath the thalamus is the hypothalamus, which, if you ever need to know, is given away by the name of hypo, meaning it is under. If there were something over the thalamus, it would chances have been called the hyperthalamus. Regardless, the hypothalamus is below this, and it is largely involved in motivated behavior. By motivated behavior, I mean any type of behavior that is directed towards or away from some type of specific stimulus, which one could argue is a lot of our behavior. An example would be hunger. When we are hungry, we are often drawn towards certain types of stimuli that will lead us to satisfy that urge. Now, it does this through the control and secretion of hormones, and is also related in this sense to the pituitary gland, which also regulates certain hormones. And this is ultimately how it is able to control our various motivational states. The amygdala, we mentioned this briefly when we were talking about Capgras syndrome, and this is heavily involved in emotions, emotional evaluation, and just managing salience in general. And this is shown in green on the diagram to the right. Next, we have the hippocampus, which actually has quite an interesting structure. And the hippocampus is heavily involved in the creation, organization, and retrieval of memories. Damage to this brain region will often result in amnesias. Some can be quite severe. In fact, there is a very famous patient in psychology, which many people hear about in their introductory courses, known as HM. And HM could not create any new memories. In fact, the extent of HM's attention was up to a couple of minutes, assuming that HM was not distracted, such that you could introduce yourself to HM and then have a partner of yours distract him by showing him an apple or a potato, let's say, and then he would look back to you and would no longer know that you had met at all. And this was ultimately the result of an accidental surgical procedure. They were trying to treat him for, I believe, epileptic seizures, and they just assumed that this part of the brain wasn't really needed, and so they cut it out. And much to their dismay, it really messed with his memory systems. On the other side, it does show you the incredible robustness of the brain to be able to have just chopped out an extra part of the brain and that HM could largely function. I mean, barring the fact that he could never function in daily life without creating new memories, but that doesn't change the fact that he could walk and carry a conversation, assuming it pertained to things he had learned before the operation. Honestly, I think that's pretty wild. Getting into the concept of lateralization. This refers to the fact that most brain structures are duplicated in either hemisphere. So to go back just for a second, oh no, it will ruin the recording, unfortunately. Sorry, you'll have to draw up the picture we've been looking at from memory or look it up in your textbook. So when we looked at the things like the hippocampus or the amygdala or the hypothalamus or even the hemispheres as a whole, they exist on either side of the brain, meaning you have left copies and right copies. And it is important to note that there are some differences in function across hemispheres. For example, language processing is largely in the left hemisphere for most people, left and right-handed. Not for everyone though. There are a select few people who it is on the right side. But this does not mean your two hemispheres are fighting with each other and somehow doing different things and only one of them is winning at any given time and ruling your world. Right? It is incorrect to say, even though this is commonly used in language, that the left side is the analytical side and the right side is creative, or something like, you must silence your left side so you can express more creativity, or you'll be able to solve your math exam better if you could just shut down your right side. Your brain does not work like this. It gets talked about a lot, but this is a misunderstanding of how the brain works. I cannot emphasize this enough. Your brain is not two separate units that are fighting with each other for control. For the most part, the entirety of your brain, both hemispheres, work very closely together to perform our coordinated behaviors. Everything from being able to stand up and get out of bed to being able to listen to and process this lecture. Since they are two hemispheres, however, they have to be connected in some form, and they are connected through what are called commissures. Now, the most common commissure that is talked about is called the corpus callosum, and this is the predominant set of connection pathways that exist between the two hemispheres. If you look at this diagram that's presented, these are represented by these green lines. I'm sorry if you're red-green colorblind and it's hard to see the difference between the green and the pink. But the green lines are the ones traveling horizontally across the image, going from one side of the brain to the other. The pink lines are the ones moving from top to bottom, represent similar white matter tracks, except these are moving from the cortex down into the spinal column and are likely traveling or carrying either sensory information coming up from the body itself or conducting motor instructions down to the rest of the body. Now, the corpus callosum is sometimes severed in the case of severe epilepsy, though this is truly a last resort, and it is only used if the epilepsy is localized to one side of the brain. If you have severe epileptic seizures that arise on either side of the hemisphere, severing the corpus callosum, as far as I know, will not help. And when it is severed, you ultimately disconnect the two hemispheres from each other. Not entirely, of course, but at least through their dominant means of communicating with each other, and this results in split brain patients, which I will talk about in a couple slides. Before getting into our talk about split brain patients, it's important to understand contralateral control. Now, what contralateral control means is that one side of your brain is controlling the opposite side of your body, such that the right hemisphere is largely responsible for processing the information from and controlling the left side of your body, while the left hemisphere is largely responsible for interpreting sensory information and coordinating the movement of the right side of your body. Now, it's a little bit more nuanced than just dividing everything in half. The common assumption would be that the right hemisphere controls the left eye and the right hemisphere controls the left eye, but it's not quite that simple. It turns out that the left portion of both visual fields, by that I mean the left portion of what you see out of either eye, is actually controlled and processed by the right hemisphere, and the right portion of both visual fields is processed by the left hemisphere, which is, again, I think pretty wild, because it means that your eyes, each of them, is split up in processing across both hemispheres. Now, you can find this in your textbook. I highly recommend looking into it some more, but this is a classic experiment done to show the effects of split brain on certain patients and their behaviors. So note in the leftmost one, they've been asked to report on what it is that they see. Now you can see that they're sitting very close to the screen, and so presumably the fork is in the right side of their visual field and the spoon is in the left side of their visual field. Now I mentioned that language is largely processed on the left side of the brain, and so it is also dealing with the right side of the visual world. So this person sitting directly in front of that screen is technically seeing both things, but when they're asked about it, it is the left side of the brain that has access to the words to be able to respond, and therefore it responds fork because that is the portion of the visual field that it is looking at, and a fork is what is present in that field. Now, going to the right side of this image, you see that this person is reaching with their left hand to pick up the object that they see. Now, here it is the right portion of the brain, the right hemisphere, that is controlling their left arm. It is also what is processing what is in the left visual field and is not aware of what is in the right visual field. Therefore, once this person sticks their arm underneath to try and figure out what it is, which they're doing by feel, they will ultimately pick up a spoon. If you had made them reach with their right arm, they would have picked up the fork. Now, there is something very important to know here. Please notice how close this participant is sitting to the screen. They're sitting so close because this is the only way to ensure that these two different images are processed by either side of the brain only. If a split brain patient was sitting further away from this screen, you would not see this effect because both items would be included in both portions of the visual field. For many split brain patients, it is not going to develop into different personalities that are running your life. For most daily processes, they will still be coordinated behavior, though they are some extreme cases. But it is not going to be like this where two entire different worlds are appearing to a person at all times. Because in real life, you could just move your head around and you would be able to look at different aspects and have them be processed by both sides of the brain. It is only in very controlled situations like this one that we are able to truly see the effects of split brain patients and what effect that has on their behavior. Once again, just having split brain patients does not mean that these people have two entirely different personalities in their head. Moving on to neuroimaging technologies, we are going to talk about 5 of them. Computerized axial tomography, positron emission tomography, magnetic resonance imaging, functional magnetic resonance imaging, and electroencephalography. Their common abbreviations are given next to them. The first is computerized axial tomography, and this is basically taking an X-ray of your brain. Here are some pictures to show what it looks like, and these are just slices at different levels, as if you took a slice parallel to the ground at different heights through your head. And using X-rays along with detectors placed all around the head, one is able to generate a three-dimensional structural map of the brain. Now, it works fairly well, and you can use these types of CT scans on other parts of the body as well. This is oftentimes when there is a broken bone or some such, you're often ordered to get a CT scan of some kind. Please be aware you probably don't want to have too many of these done over the course of your life because X-rays can be damaging to tissue. Next, there's positron emission tomography, and this actually allows us to see function within the brain itself. You'll see 3 different images here. It is not really relevant to the course that these are of normal, mild cognitive impairment and Alzheimer's disease, even though it is interesting to see the different functionality. Now, please note that there are no actual colors in the brain as are shown on this picture. Rather, these are added to the brain images later on to give an index of activity. And oftentimes, reds or hues of red are indicative to high levels of activity, while blues and greens are lower levels. So green would be in between red and blue would be the lowest levels you had recorded. Now, the way in which a PET scan is able to infer activity is by taking some radioactive glucose. Now, glucose is a single molecule sugar. It is what we break down most other sugars into when we ingest them because it is what can be processed by our cells. And the way that they're made radioactive is by using a heavy isotope of carbon. Oftentimes, I think it's carbon-14, which is added to the glucose when it is combined into a molecule, and ultimately you get a slightly radioactive sugar. This is then in a solution of some kind injected into the bloodstream, and as that radioactive isotope decays back down to a regular standard atom, it releases energy, and this energy can be picked up by detectors that are placed around the subject. Now, as the brain is going through this glucose, because one uses a lot and glucose is ultimately what drives it, we are able to infer the activity in the brain by looking at what parts of the brain are using more or less glucose. And this is how we are able to color these maps and say that certain parts of the brain are using much more or are more active than other parts of the brain. Going on to magnetic resonance imaging. So this is a more modern technology, and a standard MRI or magnetic resonance image will allow us also to see very high definition structural images of the brain. Here are some additional images presented, and again, it is not really relevant to this class, but you can see that there are these weird little islands that are present also, and these are likely to show some kind of trauma or lesion. It could even be a severe stroke. Now, The way an MRI works is it applies a very, very strong magnetic field to the participant, well, technically in the chamber in which you are inserted into the MRI. And this magnetic field is so strong that it aligns the various little protons, which are parts of the atomic nuclei, all into a certain and specific direction. After this, they pump certain radio frequencies in alternating patterns through the person. And this disrupts the alignment of these protons that are now trying to align themselves to the magnetic field itself. As you then pulse the radio, by that I mean it goes on and off, whenever that radio signal is turned off, the protons try and snap back to the alignment of the magnet. And in doing so, they release some energy, much like how the radioactive decay did, except less damaging because you're not putting radioactive material into the human body. This release of energy can then be picked up and it can be used to create this image. Now, different densities of tissue will release this energy at different rates, and this is how we're able to build such intricate maps. Now, please note that I went into probably more physical definitions of how this works than you really need to know, but it is one of those cool modern wonders of science that we're able to even do something like this, and so I wanted to give it its due credit. Because the fact that we can move around atomic nuclei and then disrupt them out of something we've forced them into and then use how they pop back to where we align them forcibly in the first place to infer the structure of something that we cannot actually see is pretty unbelievable. Next, we have functional magnetic resonance imaging, which is very much like a conventional MRI, except it, instead of just looking at protons, is looking specifically at hemoglobin molecules. Now, a hemoglobin molecule is what binds oxygen. It is what allows us to carry oxygen throughout our blood, and it is also what makes our blood red largely. Now, hemoglobin has different magnetic properties when it is carrying oxygen and when it is not. So this is, you could imagine that it has a certain sort of magnetic field when oxygen is bound to it, and it has a different magnetic field when oxygen is not bound to it. This difference can be picked up by a functional magnetic resonance imaging device, or an FMRI. And this change in blood oxygenation can be used to infer activity in the brain, much like the PET scan, except where the PET scan used sugar being used by the brain, in this case we are looking at how the brain is consuming oxygen in order to maintain its function. Basically, for any type of running of a cell, you need sugar and you need oxygen in order to generate energy. This is why these two are often used. But so an fMRI ultimately uses this change in blood oxygen content to infer the level of activity in a brain. And here is a sample image where, again, reds are indicating heightened levels of activity and blues are indicating relatively low activity. Chances are this person is looking at something right now because you can see a tremendous amount of activity in their occipital cortex. No guarantee though, they could technically be imagining or visualizing something and you might see similar activity. We'll also talk about that as we carry on. Finally, we have electroencephalography, and this is a little bit different than the last four we've talked about. So electroencephalography, or abbreviated to EEG, it works by placing electrodes onto the scalp of a person. It can also be on the hair, pushed down, and then a specialized electrode gel is injected through them, which makes an electrical connection between the scalp and the electrode. And it can be anywhere from one, which we use to detect auditory brain function in newborn babies to ensure normal brain functioning, all the way to some advanced systems right now that have up to 256 electrodes that can be stuck onto the head. This works by measuring voltage changes that are the result of clusters of neurons in the cortex firing. And we use these voltage changes to infer once more activity going on in the cortex. And this is read out as a waveform. Now, a waveform is a type of graph, and the one people are most likely familiar with is when you look at those little Richter scale monitors that we often see in movies when either Godzilla is about to attack a city or a horrible earthquake is going to happen, and you see this little wiggle go up and down, and all of a sudden it goes crazy. The other place where one commonly sees this type of waveform is in movies when you have polygraph detectors, and again you see that same firing. While it doesn't look exactly like that, it is ultimately similar, and that is how we read out EEG data. We do divide different patterns into different rhythms, things like alpha and beta rhythms, and all these mean are that different regions of the brain are firing at different rates. For example, if the brain is, or a region of the brain is in an alpha rhythm, it means that it is firing at a rate of approximately 8 to 13 times a second. Now, not the whole brain together. If the whole brain was firing at the same frequency, you would have an epileptic seizure, but rather different regions are a little bit out of sync, but they will be on these similar patterns. If I was to say the brain was, or a part of the brain was in beta, it would be anywhere between 14 hertz and 30 hertz. By that I just mean cycles per second. Now, EEG is incredibly temporally accurate. By that I mean you're able to measure changes in activation in the cortex down to a couple of milliseconds, which is pretty unbelievable. But EEG is incredibly limited in trying to figure out where that activity is coming from. It is very bad at spatial recognition, or its spatial resolution is quite bad. Now, it is also worth noting that an EEG cannot measure activity from deeper down in your brain. It is only able to measure activity in your cortex, and even that in cortical regions that are perpendicular to the skull itself. Now, there is a way to measure the electrical activity of deeper brain structures, but this requires that you cut a hole into the scalp of a person and insert very, very thin wires. This is only occasionally done with certain brain surgery operations, but is much more widely used in certain types of animal and especially rat studies. A note about multimodal methods. So as I mentioned before, EEG has bad spatial resolution and very good temporal resolution, and conversely fMRIs, which are a modern form of looking at spatial resolution, have very good spatial resolution, down to about 1 cubic millimeter, otherwise sometimes referred to as a voxel. But they have fairly bad temporal resolution, and this just means that they're only able to detect changes on the order of several seconds, and your brain can actually do a lot of things in a couple of seconds, and so this is not very good for establishing when a certain thing happened. Now, to make up for these shortcomings of either, oftentimes both of these methods can be used concurrently to examine certain types of phenomena, and this allows us to look and have very good both spatial and temporal resolution. Now, that is, of course, assuming that an institution can afford both systems, because oftentimes EEG systems, especially fancy ones, can cost upwards of tens, if not hundreds of thousands of dollars, and fMRI machines can easily go into a couple, if not more, $1 million. It is, unfortunately, one of those things where it is still very much pay to play. One last thing I wanted to talk about in this portion of the lecture is dispelling a very common myth around the brain. And there is this common thing that states that we only use 10% of our brains. And this probably persists due to the idea that if we can somehow access more of our brain, we can somehow increase our performance in some way or become greater than we are. You see this in movies and stories all the time, or even in a lot of ultimately scam self-help ideas that teach you how to unlock greater potential in your brain. This is not true. A healthy functioning human being, or any animal with a brain, uses all of its brain. I'll repeat this again. All of us who are healthy functioning humans use the entirety of our brain. What we use it for could be argued to be different, right? Some people might use it for rocket science and some people might use it for keg stands, but that doesn't change the fact that we are always using the entirety of our brain. If we were not, it would not make sense for almost a quarter of our daily calories to be spent on the brain, which is a lot of energy, especially when you consider that supermarkets and Frito-Lays did not exist until much recently. In fact, for most of our evolutionary legacy, We have been running various calorie deficits in our entire existence because there was not always an abundance of food. There would be no reason to keep such a large brain if it was not doing something. Please be aware, it is not true that we only use 10% of our brain. I'm sorry if this makes you feel like you cannot somehow magically be greater than you are, but this does not change the fact that you, one, use all your brain, but don't be too sad because you are actually able to change your brain. Whenever you learn something, you are changing the manner in which your brain connections are situated. We'll talk about this much more as we go on through this course, but ultimately what you are doing when you are learning is changing your brain. And every time you learn something, there exists the possibility of you being greater than you were before you learned that thing. Just think about how you have learned new skills. Every new skill you have learned has made you at least in some way greater than you were before, no matter how trivial that skill is. So please don't rely on some easy quick fix solution to move from 10% to 15% of your brain. That person is lying to you. But take solace in the fact that you can continue to learn new things, and that is pretty incredible. Not to mention you can probably do it for, whatever willing 80 plus years of your life. If you want to spend the energy. That is the big rub. At the end of the day, learning takes energy because it is effortful and it takes time and you are reorganizing your brain. The reason most of us stop learning is not because we don't have enough brain power. It's because we're lazy and we get distracted.